Hello students, this is CV Manoj. We have already discussed about carbohydrates in my previous lecture. Today I am going to start with proteins and amino acids. Even this is a part of biomolecules. In proteins and amino acids, we are going to discuss about the different types of amino acids, actually what is the relationship between proteins and amino acids, which is a bond which links the different amino acids giving rise to proteins. So all these parts will be discussed in this particular lecture. Let's start with it. Now, as you can see over here, biomolecules is nothing but made up of two molecules. Two molecules in the sense they are of two types. One is micromolecule and the second one is macromolecule. Micro means which is of low molecular weight substances, and macromolecules in which macro means the larger molecular weight substances. This micromolecules involves water, gases, sugars, amino acids, nucleotides, etc. And when you're talking about macromolecules, which is of larger molecular weight, they are nothing but carbohydrates, lipids, proteins, and nucleic acids. If you can see over here, this carbohydrate is nothing but made up of sugars, and this proteins is nothing but made up of amino acids. Okay, and when talking about nucleic acid, it is made up of nucleotides. So that means this macromolecule is in turn made up of this micromolecule. Now, what is protein? Protein is nothing but organic molecule which is present in almost all the living system. It is one of the fundamental bases of structures and function of life. The way we say cell is a basic function of a life, a structural and functional part of a life. Same way protein is present in almost all the living system. 50% of the dry weight of every cell is because of protein molecule. And this protein is nothing but polymer of L-alpha amino acid. That is what is the relationship between amino acid and protein molecule. So I can say protein is made up of amino acid. So amino acid is a monomer and protein is a polymer of this amino acid. Near about 300 different types of amino acids are present in nature. Amongst these 300, there are 20 standard amino acids. Now, these 20 standard amino acids, the names will be discussed later. There is one more 21st amino acid which is added to the list is selenocysteine. Selenocysteine. Cysteine is already present in this 20 amino acid. But when it is linked with a selenium metal ion, so this selenium is linked with cysteine, giving rise to the 21st amino acid, which is selenocysteine. Okay, now next, from which word the term protein was derived? Protein was derived from a Greek word called as proteos, and proteos means the first place. Berzelius, the Swedish chemist, is the name of a scientist, a Swedish scientist. He suggested the name proteins as a group of organic compounds, which are nothing but nitrogenous macromolecules made up of amino acids. Why it is nitrogenous macromolecule, you will understand in a better way if you just concentrate on the part of the structure of amino acids. Now, what are these amino acids? As I already mentioned, amino acids are nothing but the monomers and proteins are polymers of amino acids. So I can say proteins are made up of amino acids. Now, what are these amino acids? These are nothing but organic compounds, and they are made up of two functional groups. One is amino and other one is carboxyl. Now, I said that it is a nitrogenous organic compound. Protein is a nitrogenous organic compound. Presence of amino gives it a nitrogenous compound. Amino group is present, carboxylic group is also present. Now this amino group is nothing but NH2. Carboxyl group is nothing but COOH. Because of presence of amino group, it is basic as well as because of presence of COOH, it is acidic in nature. So it can be both basic as well as acidic. As already discussed, there are total 300. Amongst the 320 of them, 20 of the amino acids, they occur in the proteins as already mentioned. Now, what is next? Now, let's talk about the structure of amino acids. Each amino acid is made up of 4 different groups. These 4 different groups, they are attached to alpha carbon. As you can see, this is the structure of amino acid. This is one alpha carbon present in the center. These are the 4 different groups. That is, one is carboxylic group, this is amino group, one is the R group, which is a side chain, and this is the H hydrogen atom. Again, I'll explain you this. This is a structure of amino acid because of which protein molecule is formed. This center, in the center you can see there's a carbon atom. This carbon atom is alpha carbon atom because it is linked to a functional group, which is a carboxylic group. So here, this carbon atom is already, you all know the chemistry behind carbon. It is a tetravalent. So this tetravalency is covered by because of 4 groups. One is carboxylic group, other one is amino group, the third one is R group, which is a side chain, and the fourth one is the hydrogen atom. You cannot disturb the carboxylic group, amino group, and H as a hydrogen atom. But one can, this R can be changed. In what way it can be changed, we can discuss it later. Now, check it over here. Even this is the structure of amino acid. This is C carbon, which is alpha carbon. This one is carboxylic group, you can see over here. This one is amino group, which you can see over here. This is common to all amino acid of proteins, means this amino group, this carboxylic group, this H, they are common to almost all the amino acids of the protein. But when we're talking about the side chain, is distinctive for each amino acid because of the changes of the side chain R. The side chain is changing in each and every amino acid. So because of the changes in the side chain, it gives rise to different amino acids. Alpha carbon is between the carboxyl and amino group. You can see here, this is alpha carbon. So this alpha carbon is between this carboxyl and this amino group. That is what is the structure of amino acid over here. It's very simple. One is the alpha carbon. They are linked to the 4 group. Amongst all the 4 groups, 3 groups are constant. One R side chain remains. It differs according to the amino acid. Since it differs, there are 21 amino acids present. So the difference in the amino acid is because of this R group. Okay, next. Now actually, what is amino acid? It's just briefing out again. Amino acid is nothing but an organic molecule which is made up of one amino group, one carboxylic group, one hydrogen atom, and one specific group, that is the R group. This is attached to the central alpha carbon atom. R group, the one with specific group for each amino acid, they varies in their structure, in their size, in their electric charge, and in the solubility of the amino acid in water. What all are amino acids made up of? They are made up of carbon, nitrogen, oxygen, hydrogen. As already instructed, amino acids are nothing but the building blocks of or structural elements of protein molecule. Next, this is a basic structure enlarged as I shown it to you. Basic structure amino acids again. This is the alpha carbon atom. These are the 4 groups. One is hydrogen, one is amino group, and the third one is the carboxylic group. And these 3 groups will remain the same as it is in each and every 21 amino acid. The only one differs is this side chain. Because of this difference in the side chain, we get 21 amino acids. Fine. Now this is amino group. NH2 is the amino group. This is carboxylic group. C double bond OH is the carboxylic group. And this is the hydrogen atom. And this is the side chain, you can call it. Okay. Now, what is actually this peptides and proteins? What is the relationship between peptides and proteins? As already mentioned, that amino acid is the one monomer which gives rise to the polymer protein. But what is the one which links all these amino acids together? It is nothing but a bond. Now what is the name of the bond is the next part of our discussion. That means proteins and peptides. What are peptides? These are small small units of the proteins. Proteins and peptides are the amino acid polymers in which individual amino acid units, which are called as residues. What are residues? In the previous you saw R. That R you cannot call as residue. You can call residue in a polypeptide chain. The individual amino acid in a polypeptide chain. Polypeptide. Poly means many. Peptide means a peptide bond. But I'll come to it later. So in the individual amino acid in a polypeptide chain is called as residue. Now there are so many amino acids giving rise to protein molecule. But this amino acid, they are linked by a particular bond. That bond is nothing but amide bond or peptide bond. Okay, so each amino acid, they are linked by amide bond or peptide bond. Now, next, how is this peptide bond or amide bond formed between two amino acids? How are they formed? Just check out here. This is one amino acid. This is the second amino acid. Till now, I have not listed out the amino acid, but just out of the 20 amino acid, one amino acid is alanine, and the second amino acid is serine. As you can see, this is the alpha carbon atom. This is alpha carbon atom, and this is the alpha carbon atom. This alpha carbon atom is linked with four groups. One is H, which is constant. One is amino group. One is a carboxylic group, and the other one is CH3R, which is keeps on changing according to different amino acids. Now this is serine. In this serine, this is the alpha carbon atom. This alpha carbon atom is again linked to the four groups. One is H, other is amino group. The third one is carboxylic group, and here the R is CH2OH. This is serine. So what I want to show over here is the two amino acids, they are linked together to give rise to, I can call this as a dipeptide, because what you can call two amino acids are involved in this. Now there is a bond. Now this is one amino acid and this is the second amino acid. This is one amino acid. Which amino acid is this? Alanine. Which amino acid is this? Serine. So this alanine and serine, they are linked by each other by means of a peptide bond. You can see over here, C double bond O NH. This is nothing but a peptide bond. So this C double bond O is because of one amino acid, NH is because of the other amino acid. An amino group, that is this amino group from one residue forms an amide bond with the carboxylic group of a second residue. That means here you have one amino acid, here is a second amino acid. Both the amino acids are coming together by means of this peptide bond. So how is a peptide bond formed? An amino group of one residue. One residue means what? Amino acid. One amino acid in a polypeptide chain. That is what is a residue. So amino group from one residue forms an amide bond with a carboxylic group of a second residue. So one residue is this, second residue is this. So amino group of this one residue and the amino carboxylic group of this residue, they come together and they give rise to what we call it as a peptide bond or amide bond. So this is nothing but peptide linkage, or you can call peptide bond. When you talk about carbohydrates, in carbohydrates you have glycosidic linkages. But when you talk about proteins, you have peptide bonds or amide bonds. Next, again the same peptide linkages. Two dipeptides can result from reaction between A and S. A is alanine, S is serine, depending on which carboxylic reacts with which NH2 we get. The long repetitive sequence of NCH C double bond O atoms, NCH C double bond atoms that make up a continuous chain is called protein's backbone. Peptides are always written with N-terminal amino acid, the one with free NH2 group on the left, and the C-terminal amino acid, the one with the free CO2H group on the right. That means one on the left hand side you have the N-terminal, and on the right hand side you have C-terminal. So a particular peptide, polypeptide chain has got two terminals. One is N-terminal, other one is a C-terminal. One N-terminal is because of the free NH2 group, and the other C-terminal is because of the free CO2H group. Now, there are some abbreviations of this alanine serine. Alanine as an example. There are some and many other amino acids which we can take. Just for simplicity, I have used this alanine, alanyl and serine. Alanyl is abbreviated as ALA and serine is SER, or one word short form or abbreviation is A for alanine, S for serine. So the linkages between the two is because of the peptide bond C double bond O NH. On the left hand side you will have N terminal, on the right hand side you will have C terminal, and that means N terminal is for because of free NH2 group and C terminal is because of free CO2H group. That is, this is N terminal and this is the C terminal. Next, these are the list of the different amino acids. Over here you can see the different amino acids like alanine, arginine, aspargine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, then methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine, aspargine or aspartic acid, glutamine or glutamic acid. So these are the list of 21 and 20 amino acids, the 21st in the cysteine. Now, each one of this has got 3-letter abbreviation and 1-letter abbreviation. That means you can see over here alanine, the first 3 alphabet ALA is its 3-letter abbreviation. Like that, there are so many other amino acids. The first 3 words are their 3-letter abbreviation. When I'm talking about 1-letter abbreviation, the first one. Now you can see alanine A, so A we have. But arginine is also A. So we cannot give it that as A because it will create a confusion. Which one is alanine and which one is arginine? So arginine is given R. So like that, aspargine, the short form abbreviation is capital N. Aspartic acid, it is D. Cysteine, it is C. All these are capital letters. That means these are the 3-letter abbreviation. These are the 1-letter abbreviation. It's nothing. It's not like the first alphabet is a 1-letter abbreviation. It's not necessary because the first letters are common in almost most of the amino acids. So to be on simpler side, alanine A is given A as a short form, arginine is given R as the short form. Okay, now what is the next? Now, what are the different types of amino acids on the nutritional aspect? There are 2 types. One is essential amino acid, which is indispensable amino acid, and the other one is non-essential amino acid or dispensable amino acid. Now, what do you mean by essential or non-essential? Essential, something what is from the name itself you can understand that something is required by our body. Non-essential means not that much required by our body. But how will explain in a better way? Essential amino acids are not synthesized by the body. The essential amino acids are not synthesized by the body. They should be supplied through diet, are the essential amino acids. I repeat, essential amino acids are the ones which are not synthesized by the body. They should be supplied through the diet. They are required for the proper growth and maintenance of the individual. Now, what are the list of the different essential amino acids? The list are arginine, valine, histidine, isoleucine, leucine, lysine, methionine, threonine, tryptophan, phenylalanine. So these are the list of different essential amino acids, the ones which are not synthesized by the body. Since they are essential, we have to supply them through the diet externally. Now, what is semi-essential amino acids? Semi-essential amino acids are the ones which are synthesized by the adults, but they are not seen in the growing children. So, semi means half. They are synthesized by adult, but they are not seen in the growing children. For example, arginine and histidine. So this is a molecule, the formula of arginine, and this is nothing but histidine. As you can see over here, which is a carbon atom over here, alpha carbon, can you tell me? Now this is the alpha carbon which has H1 group, carboxylic CO minus, another one is amino group, and all this is the side chain, you can call R group is the side chain. So this is the N terminal and this is the C terminal. Even in this histidine you can see this is the alpha carbon atom which has got one H, the other is amino and the other one is carboxyl. Now in this case you can see this is a side chain. Now next, What is non-essential amino acid? Non-essential amino acid is the one that can be synthesized by the body and need not be consumed through the diet. Is a non-essential because it can be synthesized by the body. It's there in the body. Even though you don't take this amino acid externally, it's well and good because it can be synthesized by the body. What are the different amino acids which are non-essential? Glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, proline, tyrosine. Okay, so these are the non-essential which can be synthesized by the body. That's the reason why it need not be consumed externally. Now, when we are talking about the 21st amino acid, which is selenocysteine, if you can go through the slide in which I have given the different amino acids, in that you can see cysteine is there. Along with cysteine, one selenium is attached to the cysteine. So it gives rise to 21st amino acid, which is selenocysteine. This is present during protein synthesis, and they are created through post-synthetic modification. Post-synthetic means after the modification of amino acid residue. Okay, and this is what is the selenocysteine structural formula. Again, is the same C alpha carbon. This is one H hydrogen atom. This is the amino group. This is a carboxylic group, and this is nothing but our the side chain. So this is selenium over here. That gives its name selenocysteine. Okay, now what is next? Amino acids as ampholytes. These amino acids, they can be called as ampholytes. Reason is they have both acidic group as well as basic group. Acidic group means it has both carboxylic group COH. Basic group means it has NH2 group. They can donate or accept a proton, hence they are known as ampholytes. They can donate a proton as well as they can accept a proton. Proton means H+. So they are called as ampholytes. Now this form, you can see over here, this form of this amino acid is Jupiter ionic form. Reason is there is both presence of proton plus positively charged as well as negatively charged species. In this Jupiter ionic form of amino acid, they have both positively charged species as well as negatively charged species. The positively charged species is nothing but NH3+, and the negatively charged species is nothing but C double bond O minus. So the one in which you have both plus and minus is a Jupiter ionic form. Okay, it's a Jupiter ionic form. Now here what is happening, this is COOH group, and this H is released, and they are, they are captured by this nitrogen of this amino group. So when H is released from here, you have what? COO minus like this, and one H is captured by this nitrogen atom, so it becomes NH3+. And that's why we get Jupiter ionic form, in which you have both negatively and positively charged species in it. This is a Jupiter ionic form of amino acid, in which you have the negative, that is a carboxylic group, and the positive, that is the amino group of the amino acid. Okay, now what is this actually? This is already discussed about Jupiter ionic, which is neutral. Why it is called as neutral pH? The reason is it has got both positively charged as well as negatively charged species, that is Jupiter ionic neutral pH. Now in this case you can see there is only one positively charged species over here, and here you have C double bond OH. This is low pH. Why low pH? Because it is full of protons are present over here. More protons are H+ is present, it goes to more acidic side, and if it is acidic side, it is of low pH. But when you talk about this, you have CO minus over here. One H is lost from here, one H is lost from here. That is less amount of proton you have in this. That means it will be less acidic and more basic. And since it is more basic, you can call this as high pH value. That means in strongly acidic pH, amino acid is positively charged. Strongly acidic pH, very strongly acidic pH, this is strongly acidic pH, so the amino acid is positively charged. In strongly alkaline pH, that is this is alkaline, where you have less amount of H+, so in strongly alkaline pH, it's negatively charged. It means amino acid is negatively charged species. But the one in which you have both positive as well as negative, that form of amino acid is called as Zwitter ionic form. Okay, now what is the next slide? Next is, what is isoelectric pH now? Isoelectric pH is a pH at which molecule exists as a Zwitter ion or dipolar ion and carries no net charge. Here the net charge is 0 in isoelectric pH. In Zwitter ionic form, the net charge is 0 because they have both positive as well as negative charge. They are electrically neutral at isoelectric pH. They means the molecule, the amino acid, they are electrically neutral at isoelectric pH. It is a pH at which the molecule exists as a Zwitter ionic form or dipolar. Di means two, polar means they have positive as well as negative charge species. Both they have. So they are called Zwitter ionic form, in which they are neutral in form at the isoelectric pH. It's a pH in they are neutral because they have positive as well as they have negative charge on them, which is a Zwitter ionic form. Next is the different isoelectric point they have shown over here. This is a Zwitter ionic form which is neutral, in which you have positive as well as negative charge. Now this, after the release of H plus from here, this is the anionic form in which you have a minus charge over here. And at the same time, this is a cationic form in which you have HC double bond OH and NH3 plus over here. This is a cationic form. But this is a Zwitter ionic form in which you have both COO minus and NH3 plus. So this is a neutral or the Zwitter ionic form. Okay, anionic form is the one in which you have COO minus. Here the pH is 13. That means they are basic in nature and the net charge is -1. Here pH is 7. They are neutral and the net charge is 0 because they have plus as well as minus over here. Now here what is the pH? pH is 1 and because it is acidic. So net charge is what? +1. Okay, because it has got NH3 plus. Now what is next? Next is the structural organization of proteins. There are three types of proteins. One is primary structure, that is, there you can see this is a notation for primary structure, secondary structure, tertiary structure and quaternary structure. So these are the four types of protein. This is structural organization of proteins. One is a primary structure, second one is a secondary structure, third one is a tertiary structure and the fourth one is a quaternary structure. As you can see over here, this is a primary structure which is made up of the amino acid chain. This is a secondary structure. You can see some helix formation, alpha helix formation, curled steps you can see, a rotation of the steps. So that is the alpha helix, which is a secondary structure, and this is a tertiary structure which is nothing but of mass, what is called a part of this is seen. So many secondary structures gives rise to tertiary structure. That is so many polypeptide chains. And what is quaternary structure? So many tertiary structures gives rise to quaternary structures over here. Okay, that means this primary structure gives rise to secondary structure gives rise to tertiary structure gives rise to quaternary structure. Or you can say secondary structure is because of primary structure, tertiary structure is because of secondary structure, quaternary structure is because of tertiary structure. So these are the different structural organization of proteins. Now what is next? Next is the primary structure. It's a chain of amino acid residues which is made up of covalent bonds such as peptide bonds and disulfide bonds. So it's a primary structure which is made up of polypeptide chain. The most important element of primary structure is a sequence of amino acid residues. Now what is secondary structure? Secondary structure is nothing but the stable arrangement of amino acid residues giving rise to recurring structural patterns. That is, a primary structures which are recurring itself giving rise to secondary structure. Now what is tertiary structure? Tertiary structure is nothing but three-dimensional folding of a polypeptide chain. That is what is a tertiary structure. Means it is a three-dimensional structure of a polypeptide chain is nothing but tertiary structure. Now what is quaternary structure? Quaternary structure is a protein which is having two or more polypeptide subunits, and the arrangement of this means they have two or more subunits. Means what? Subunits of polypeptide chain. That is, they have made two or more subunits of this tertiary structure. That is nothing but called as quaternary structure. So we have four types of protein molecules: structural aspect, primary structure, secondary structure, tertiary structure, quaternary structure. Hope so you have understood the structure of proteins and amino acids. Meet for the next lecture. Thank you so much.